Thank you so much for the introduction and for providing the opportunity to present my work. Um, uh, in the next 15 to 20 minutes, I'm going to provide what I hope will be a somewhat different perspective into applied bacteriophage research. And now I know this is this section on molecular genetics. This is not at all related to molecular genetics, but probably related to phage therapy or phage biocontrol. As I said, applied phage research. And so I'm an engineer, a different kind of phage researcher than what you might expect. And bacteriophages are very attractive for bioengineers such as myself, and I'll tell you why. So what you might ask makes bacteriophages interesting for an engineer over all the different hosts of nanoparticles that engineers have created. And so there are three main characteristics that you're all very familiar with that makes bacteriophages very attractive for us that we have not been able to reproduce in synthetic materials. So host specificity, the diagram that we all know by heart. You might argue that specific recognition is a characteristic that is monopolized by nature. And so we haven't been really able to reproduce this in engineering built materials. However, bacteriophages specifically, they offer a very different brand of specificity down to the strain level. The other characteristic which makes bacteriophages attractive is self-replication. So during the replication cycle, theoretically one or a few bacteriophages are able to produce hundreds and hundreds. So that self-replication on spot is unmatched. And this is what we're all used to seeing as the clearings on the plate. Now that self-replication leads to rapid evolution, which is again, very attractive if you're designing engineering systems and materials. And last but not least, diversity. Now this diversity is in shape, it's in size, it's in surface chemistry, and it's in modes of replication, of course. And all of this is unmatched. Now, needless to say that the characteristics that make bacteriophages exceptionally attractive for a bioengineer are the ones that make it extremely challenging to work with as well. Because if I work with a nanoparticle, and I have done a lot of that, I don't need to worry about it changing from one process to the other or replicating on the spot. So yeah, both very attractive and challenging. So let's jump right in. What have I done with bacteriophages from the applied perspective? Now, when it comes to building materials with phage, not materials doped with phage, entirely with phage, the filamentous phage is very interesting. Now, the picture that you can see here is not to scale, of course, but you can see a filament. It's long and it's very, very thin. So very high aspect ratio. And what this shape does is that under certain conditions, you can lead it to organize and self-organize. And you can see, so what you can see in orange, the fibers, these are actually filamentous phage. This is M13 phage, which the host for M13 is E. coli, which we've replicated, which we've propagated in very high concentrations. And a confined medium, you can see that they start to assemble and form these very ordered structures. Now, this is an image produced with an atomic force microscope. And the areas that are lighter, those are the P3 protein, which is the P3 proteins, larger, they're sticking out. Now, what we've done with the M13 phage is that we pack them into a gel. The gel that you can see here, the goldish, yellow goldish looking puck here, that's entirely made out of M13 phage. slightly dried out, but entirely made out of M13 phage. And if you look at this with a polarized microscope, that order, that self-assembly manifests itself into a property called this iridescence. So light is reflected from different angles, depend on how you look at it. And you see this beautiful color that shows up. That is very important because a lot of the, this is what, it's displaying similar property to liquid crystals, which you can find in some screens, and in a lot of biosensors and bioassays. So pair that with specific recognition, that's very powerful. Now, the reason that you see this color, as I said, is order at the micron nanoscale. So when we zoom in with the scanning electron microscope, you can see the ordered layers. And if we tear this up and look at it a little bit further with a little more detail, I hope you can see the lines here. These are all phage fibers, bundles of M13 phage. And again, if you rough it up a little bit further and look inside, I draw your attention to the scale bar here. Again, you can see bundles of bacteriophages. The black and white arrows are pointing to bundles of phage. Now, these are much thicker than one single phage nanofilament, but still, it's these are bundles of bacteriophages. Now, the interesting property of this puck is that it absorbs a lot of water. The diagram that you can see here shows you the swelling ratio, or the amount of water absorbed by weight. So you can see that depending on the phage concentration and the crosslinker, you're getting about 1,800 times, close to 1,800 times weight absorption. Another interesting property of the gel, which our hypothesis is that it's brought about by the chemistry that we've used, is autofluorescence, which is troublesome in some cases and very attractive in other cases. Now, the four images that you can see here, the black one is just bright field, nothing. This is the gel under a fluorescence microscope. And the other three are autofluorescence observed at different wavelengths. And this autofluorescence can decrease, will decrease as the gel is it breaks apart with proteolytic enzymes. And you can imagine injecting this in the body and following it with tracers, and the autofluorescence diminishes as the gel is breaking apart. So that's also a powerful characteristic. And last but not least, which this was something that we never expected, was when you break these gels apart. Now, what you see here, the two pucks, different concentrations, we tore them apart, brought them together in a calcium environment, and they fuse back together. So that's self-healing. So the gel has all these properties plus self-healing. And you can see this is, again, under an electron microscope, this is what the self-healing looked like. But all of that would be nothing if the phage, well, I wouldn't say nothing, still very attractive, but it wouldn't be as interesting if the phage was inactive. It would be just acting as a nanomaterial. But what made this work very exciting was that we found out that at least a proportion of the phage inside these gels remained active. So we were able to repropagate that phage, and we were able to have the phage bind to the host bacteria, as you can see in this illustration here. And so this work was published in Chemistry of Materials, and this is what my students chose as the illustration to show. So these are phage bundles that sort of fade into that liquid crystal. And so the idea, this was the genesis, this was the start of our work on phage-built gels. So you pack these bioactive nanofilaments into a material. They retain their activity, but at the same time, they exhibit properties that come with the chemistry part of it. Part of it is because of the phage, part of it is because of the chemistry, and part of it is because the structure, the macro and the microstructure. And so from that point, we moved on to decreasing the scale, making it smaller. And so we started with honeycomb structures here. And so honeycomb structures immediately brings into mind, the honeycomb. And that structure is very attractive for engineers because it exhibits a range of properties, as you can see on this slide. So what we did was we made honeycomb structure through a method called breath finger method. And we made a mold. And inside of that, we poured proteins, and later you'll see phage. And the structure that you can see at the bottom here is actually with proteins. What you can see, the very first diagram is the small spheres, that's Staph aureus. And because of the micro and nanostructure of this material, Staph aureus doesn't stick to it as much as the flat surface. So this is not because of the phage, but because of the micro and nanostructure. So again, combine the non-specific, decreasing the non-specific adsorption with the specific binding of phage, which is, again, that's very powerful for designing non-antifouling surfaces. And so this work was published in Chemical Communications, and it led the path to really the final part of the story here, which was now moving on to making use of the hydrogel plus the manufacturing or method that we'd made, which led to making microbeads, microgels with bacteriophages. And in the first diagram, you can see, the first image, you can see the mold with the tiny microgels inside. And the second one, zoomed in, you can see the actual microgels made entirely of bacteriophage. And if you zoom in a little bit further, you can see the nanofibers and the nanofilaments. And so this is all inside 1 by 1 centimeter mold, which is flexible. And if you remove the microbeads or microgels of phage from the mold, you can actually have freestanding microgels, which again, you zoom in, you can see the nanofilaments that are making up this microgel. Zoom in a little bit further, and you can see an alignment which is far beyond what we could achieve with the bulk size. Remember the puck that I showed you in the second or third slide? This is much, much smaller if you look at the scale bar. And so the alignment is so much beyond what we had seen in the bulk scale. And so decreasing the scale does that. So decreasing the scale allows us to get much better alignment. It allows us to increase the surface area, of course. And what we did with these microgels, of course, decreasing the scale allows us to now pack these into a suspension, pack these into a spray. Suspension can be sprayed, bulk gel can't. And so you pack these into a spray, and we sprayed it on of course, a bacterial lawn, and you can see that we saw a lysis zone with the patch and with the actual microgels. Not only that, we saw a significant decrease in titers of E. coli 0157 when we pack the gel, not with M13 only, but with E. coli specific 0157 specific phage, because of course, needless to say for this audience, M13 is not a very virulent phage, but the phage that we used here definitely was. And so the diagram shows you, and I know that I'm running out of time, so I will wrap up. So what the diagram shows you is the microgel with the phage that is still active and that can infectious bacterial host. And so what we did was we demonstrated the application by now spraying these gels on fresh produce and on meat, and showing the drastic decrease. And now the lettuce and the meat were artificially spiked with 0157, showing the drastic decrease in the concentration of the bacteria that infected the meat and the lettuce. And so what you might ask is the difference between just packing phage into the gel, into the spray. It's really the fact that you're preserving the bacteriophages in a hydrated microbead. So these microbeads are actually gels, they're hydrated. Not only that, you're packing so much higher concentration into one microbead than you would into a spray. So what I'm going to do at this point is, I had a few more slides not related to phage-built gels, but in the interest of time, I'm going to stop here and take a few more of your questions. But before that, I would like to acknowledge the funding agencies, the people who actually did the work, which are my students, collaborators, and of course, what you can see here are the two little rugrats that are tearing my office apart. Part of the reason that I couldn't be here in person, there in person with you. With that, I'm going to stop talking, and hopefully I can take your questions. Thank you.
Thank you very much, Zeinab. That was very beautiful, the way those phages pack in the gel like that. Are there some questions from the audience? So maybe while people I'll start their questions while people start to get their thoughts in order. Because you get those the filament structures that you showed, and then you said well, I didn't quite get what you meant right at the end when you said you were actually trying to use a different phage as well to pack that into the hydrogels. Does that also form what kind of structure? Do you get an idea of what type of structure you get from a non-filamentous phage?
That's an excellent question. So the liquid crystalline properties are really tied to that high aspect ratio. Okay. Now, our theory is that doesn't need to be just filamentous phage. Now, with the phages that we use, those are we're really going with high lytic activity because we wanted to destroy 157 on the food, but they are not they're not filamentous, of course. They've got a short neck, and so we don't get structures like that with those phages. But we have hypothesized that with the long, more flexible necks, we might get the same structures. We'll get the packing density, a high packing density, but you're not going to get the liquid crystalline.
Okay. So
Janet has a question online.
Oh, sorry. Yeah.
Amazing talk. Thank you so much. And I was wondering whether the phages are uniformly distributed within the hydrogels, and how stable are they within the gels?
So the uniform distribution to an extent. In the bulk gel, we have been able to see uniform distribution. In the microgel, we're limited to what we can see with the SEM. Right. And so what we see is, yes, we see a little bit more concentration on the surface, but, and this is typical of distribution of nanoparticles inside a microgel. So if you just made a gel and pumped the nanoparticles in it, that's what it would look like. And now I'm not talking about the filamentous. The filamentous is uniformly distributed. I'm talking about the lytic phage that would add it to the filamentous phage for 0157 biocontrol. So that's one thing. That's part one of your question. Part two of your question about the stability. We have been able to see stability to desiccation. So they're much more stable towards desiccation. We've actually tested that. So just sprayed phage on a surface, the same phage that we use for E. coli. We sprayed that on a surface and then sprayed our gels on a surface. And 2 days, 3 days down the road, assayed the surface. And of course, you would expect that there are, with phages that are not desiccation resilient, that the infectivity would be decreased quite a bit. And with the microgels, they're not. So that preservation is really important. Keeping them, just as you would with phages, in suspension, in the fridge, comparable. So we haven't been able to see a difference.
Yep, question from the audience.
Hi, it's Erica from University of Nottingham. That was a fabulous talk, thank you. I was just wondering, you've been talking about like spraying the beads onto salads and onto meat and things. Would you foresee an opportunity at any point to sort of potentially combine phages into like a film almost, which could potentially be used for like a human safe film that could be used in medical applications in future for like, say, treatment for burn victims and things?
Thank you for that question. So when we first started the work, if you remember, I showed you first a microbiome patch before the actual phage microgels. That was the whole idea. That's what we're going to do. But then the pandemic hit, and it was very, very difficult to tie a PhD student's fate to animal work, which we had in Canada. We had a lot of restrictions. So we moved on to food. That was basically, so the idea was to take the patches and put them on a surgical wounds in our case. But that really is the next step, which is what we're planning to move on to, and which was the original plan. That's why we tested the patch itself and not the microgels. We actually tested the lysis activity of the patch as well. But yes, thank you for the question. This is the next step.
Okay, well, thank you very much, Zeinab. Again, that was a really interesting talk. And so thank you.
Thank you.